Welcome back from your break. This session will focus on computational genomics research use cases. We have a series of exciting speakers who will share their experiences with Amazon Web Services, Google Cloud Platforms, and Harvard University. Please enter all questions in the Q&A function of your Zoom window. If time permits, I will moderate Q&A for each speaker after their presentation is complete. Our first speaker is Ankit Malhotra. Ankit is the Biomedical Research and Life Sciences Lead on Amazon Web Services Research Team, AWS. At AWS, Ankit helps lower the barrier for biomedical researchers to build solutions and to do their research using cloud computing. Ankit has a very impressive bio, which is detailed on our webpage for today's event. We are thrilled to have Ankit with us today to discuss Amazon Web Services as a research use case. Please welcome Ankit.
Hi, thanks, Kimberly. And I hope everybody can hear me. I'm hoping so. All right, so yeah, thanks, Kimberly, for the kind introduction. And hello, everyone. Also, thanks, Dr. Norris, for setting the stage. Here, we have a great budding relationship, ever-growing relationship with the National Institutes of Health, various activities going on, and I'll briefly touch on the STRIDES initiative as well towards the end of my talk. Today, I wanted to basically discuss with everybody present about how AWS is helping transform biomedical research, the various services that we have, and probably get into some use cases towards the end on doing biomedical and genomics research with AWS. I always want to get started by showing off this slide, which is the scientific method that most of us are, or all of us are familiar with, which is science basically starts off with a person asking a question, leads to hypotheses, you do experimentation, you get some results, you refine your model, and this goes on and on in this loop of innovation. In green on this slide here is where AWS can help you accelerate and innovate faster. We provide you with the access to critical data sets so that you can go from question to the hypotheses. We help provide services and solutions so that you can accelerate, automate your experimentation process. We help you containerize your workflows, share workflows with other folks so that you can replicate their experiments, which helps solve one of the other challenges facing the field today of reproducibility. And then there are services and solutions available for you to be able to share your work with the rest of the community to basically build things together. And then with analytical services and machine learning services that AWS provides, we help you innovate and come up with new ways of thinking how to do biomedical research. There are several challenges that are facing the field today. It all starts off with biomedical research and genomic research starting to become extremely multimodal with ever increasing data volumes. Given the extreme advances in technology that we had, we are generating data at an amazing pace. And we are also starting to integrate across multiple data sources, right? So it's no longer a single data type that somebody's evaluating. You get DNA, RNA, epigenetic data, you get data from patient, from EHR systems. They're all starting to come and integrate together into helping answer the questions in biomedical research. Research is also becoming increasingly collaborative, right? It's no longer that one lab sitting in an institute, all working in their own sort of silo. There are more and more examples of large collaboration, large consortiums coming together. And the All of Us program is an excellent example of that, right? Where NIH took the lead on creating this very large consortium for generating population level genomic data and clinical data for setting the baseline for helping develop new therapeutics. The way the research is done today in our academic centers and research institutes, it's a lot funded through the various funding agencies, and that funding / publication cycle that everybody of us is familiar with makes research increasingly resource bound. Come that deadline for a particular conference, everybody's looking to try to get on the cluster. Everybody's is becoming bottlenecked by the on-prem, and that's where I think a lot of people face challenges, even with very well provisioned on-prem resources. And then, as I described earlier, research workflows are starting to become extremely heterogeneous. There is no one way to do things. There's many ways you can accomplish the same task. And where AWS sort of starts to help, it all starts off with our global footprint. AWS does have the widest infrastructure comprised of 77 different availability zones, with each in 24 different geographical regions. And there are 5 more regions coming up. So this is a very vast network that connects the world together and brings the researchers together with the government agencies to get you access to data and be able to do your work together across the world. So I mentioned earlier, it's about science and not servers, but sometimes when you're waiting for that data to get processed, then yeah, the queue is just becoming longer and longer. It does become about compute. So AWS with our EC2 service provides the broadest set of compute instance types that is available for researchers to use. And they're all divided into general purpose, compute optimized, memory optimized, accelerated computing, and storage optimized. And then we are starting to generate these specialized instances for you to rethink how to be able to do your research in the cloud. One of the examples is the AWS Graviton processors, right? So these are custom AWS silicon with 64-bit ARM cores, and they have targeted optimizations for you to be able to use them in the cloud. What the Graviton series of AWS instances allows you to do, it allows you to rethink, reimagine how you can sort of use the ARM infrastructure to accelerate your research to be able to get better cost performance out of it. So here are 2 links, 2 blogs that we published recently, one talking about a specific benchmarking approach that we took one example, in this particular case, BWA alignment for genomic data, and we did the analysis across many different instance types to show how the Graviton, if you recompiled the BWA software to run on the ARM chips, that lets you get to the result faster and cheaper. Similarly, working with Jason Chin, we took the Peregrine genome assembler, which is a very compute-intensive task, and we compiled it, recompiled it for the ARM instances to be able to run on Graviton 2. And once again, you see those cost benefits, the cost and performance benefits by just shifting your architecture to these Graviton 2 instances. Similar example of how AWS is helping innovate in this space is our relationship with Illumina, right? So the Illumina Dragon Suite, I think everybody is aware of, anybody who's working in genomics is aware of what Dragon provides. And working with Illumina and the FPGA F1 instances that AWS provides, you can accelerate the entire Dragon Suite to be able to run faster in a cost-performant manner in the cloud. And then another example of this is our work with NVIDIA, Parabricks, which is a suite of genomics tools that lets you do alignment, variant calling, joint genotyping, all kinds of stuff using the AWS graphic board instances. Once again, setting the standard for being able to reimagine how you do your work in the cloud using the latest and greatest hardware that's available out there. And what it allows you to do, and this is where I was going towards, is that it starts to redefine what is a compute cluster, right? So compute clusters in the cloud become fit for purpose. Let's say, take the example of a DNA variant calling workflow, which has many different steps: primary analysis, secondary analysis, tertiary analysis. You can set up your infrastructure in the cloud so that when you're doing certain tasks, you can use the infrastructure that is suited for that task. So let's say primary analysis using the Dragon Suite, and you do use FPGA F1 instances and get to the result faster. Similarly, now you're going from primary to secondary analysis, compute bound. You spin up a CPU cluster, maybe using the Graviton processor. Now you're in the tertiary analysis stage, and this is where there's a lot of machine learning, data analytics, image processing happening, and you switch again. You spin up a GPU cluster, and you're able to, once again, accelerate and be able to do your science faster. Compute clusters in the cloud become elastic, right? You're no longer paying for a large infrastructure when you don't need it. Take the example of that bioinformatics student who's writing a program. In the morning, he spins up a small development cluster. He does his development. He's ready to test it out. In the afternoon, he spins it up. He runs it across tons of samples, gets the data out, and now he's just analyzing the data. He doesn't need the cluster anymore, so the cluster just goes away. It also, by going with a cloud platform, allows you to sort of think of compute as being infinitely scalable, right? With the vast infrastructure that AWS provides or other cloud services provides, you can literally spin up production level workloads. In this particular example from 2019, the researchers were able to spin up 51,500 GPUs for a single compute. And across the 2 hours of experiment, they were able to achieve performance that was almost close to the number 1 HPC in the world, right? So that talks about how the cloud could be thought of as infinitely scalable. Where the infrastructure will support when you need to go fast, when you need to go wide. Let's talk about data access, because this is the other big challenge facing the field today is large amounts of data is being generated. How do you get to it? Right, so AWS provides a vast array of services for you to share your data, manage your data, get performant access to your data, right? So I won't go through all of this. I'll share the link where you can get more information on, for example, AWS S3. The particular initiative or program that I wanted to talk about is the AWS Open Data Program, right? So this is a sponsorship program that AWS runs, which covers the cost of publicly available high-value cloud optimized data sets, especially in the healthcare and life sciences field, for it to be shared by the community, be readily accessible to the community to use. And in here, I just give you a few examples, right? TCGA, ENCODE, 1,000 Genomes, the Microbiome Project. And most recently, as Dr. Norris mentioned, we just had a collaboration with NIH, NCBI, to onboard the entirety of Sequence Read Archive, which is about 35 petabytes of data, onto the AWS Open Data Program, so that now this data is available for anybody to use free of cost, right? And at last count, I think we had about 250 plus data sets hosted on AWS S3 as part of this program, with about 80 data sets that were sort of healthcare, biomedical research, genomics focused. Let's talk about reproducibility, because that's one of the other challenges that's facing the field today. There was a series of articles that I read that back from 2015, 2016, that talked about reproducibility becoming the biggest challenge in the field today, when context matters, right? A particular lab publishes a paper, they use a particular kind of software tool, a particular version of it with certain number of parameters, and the other lab is just not able to replicate it. So this is where, I'm sorry, I did have a slide on it. So this study by Amgen back in 2012, and then subsequently another study in 2016, talks about how very few of the published papers were actually reproducible. So this is where AWS can help you sort of achieve reproducibility by containerizing your workflows. By containerizing workflows mean you set the parameters you want, you set the versions you want, you can create a virtual image out of it, and then you share that virtual image with the rest of the community. And whether it's the Elastic Container Service or the Elastic Kubernetes Service, that's what allows you to manage, deploy, schedule containerized applications on AWS. And specifically, when it comes the time to use these containers that somebody else has provided or you have developed, we provide services like AWS Patch, right, which is a managed service for container-based jobs. You can take a containerized workflow and run it across a whole host of data sets or samples that you want to process in a cost-efficient manner using AWS Patch. And we also support the community workflow description languages, for example, Cromwell and Nextflow, to run optimally on AWS. So let's get into some use cases. I think I'm ahead of time. I might be rushing too fast. This is one example of a blog that one of our subject matter experts published recently, which talks about how you can do tertiary analysis of multiomics data, in this particular case from the Cancer Genome Atlas on AWS. And I think a lot of us face this challenge right now, especially in the space of cancer genomics, where there is TCGA data available. Now, how do you get from having all of this data available on mutations, expression levels, copy number across the genome? How do you bring that all together into a single queryable instance, right? So this one particular infrastructure shows you how you can take data from TCG open buckets available on S3 in the Open Data Program, ETL them using AWS Glue, write the data into a Parquet format, basically a format that allows for easy queries, standardizes it. And then using Amazon Athena, you build an interface that can let you run the queries against this data, right? So the researcher using a VPN connection can log in to spin up an RStudio server on EC2 and be able to run their queries against this queryable data now for asking the questions that he wants to ask. AWS works with a whole bunch of a large number of researchers at many different institutes, right? So across the spectrum, whether it's pharma, working with Bristol Myers Squibb, Merck, Takeda, our research institutes, Harvard Medical School, University of California, Santa Cruz, Children's Hospital of Philadelphia, Baylor College of Medicine, and then in biotech, right? The great work that Moderna did in the current pandemic, they were able to accelerate the production of the Moderna vaccine, and part of it was enabled through working on AWS. Here is one example of a particular research institute, in this particular case, Fred Hutch Institute in Seattle, that basically used AWS, moved their infrastructure, and using EC2 with Nextflow workflows running using AWS Patch, were able to accelerate their particular use case to go from to basically analyze some 15,000 biological samples that would have taken them some 7 years to process and be able to process them in under 7 days. It allows you to speed up that experimentation process, get to the science faster. Another particular use case I wanted to talk about is the Munich Leukemia Lab, right? Munich Leukemia Lab is one of the leading institutions in the world for leukemia diagnostics and research. And MLL uses Amazon infrastructure, especially the FPGA-based F1 instances, to accelerate this genomic data processing, and then using S3, be able to work on that data, going from processing patient data in under 3 hours. Wanted to also quickly touch on using the AWS AI ML services for accelerating, and how you can start to think about innovating in the cloud, right? So this is the AWS machine learning stack, which is the broadest and most complete set of machine learning capabilities. And the way this is organized, it's available at 3 levels for the various kind of use cases that we see. At the framework and infrastructure level for those machine learning scientists who just want access to TensorFlow or PyTorch to be able to build their machine learning solutions, AWS supports all of them. And then for the next level up, for the data scientists who wants like integrated environment to be able to do their machine learning tasks, yeah, Amazon provides SageMaker. And there's a whole slew of services available through SageMaker for you to, for example, aggregate, prepare the data, use Jupyter notebooks and for visualization and for processing, train your models, tune the parameters, get access to control. And then one level of abstraction above it is the AI services. And the 2 ones that I wanted to sort of highlight here, I mean, there's many of them available, the two particular ones that I want to highlight is Amazon Transcribe Medical, Amazon Comprehend Medical. And so here is one particular example of a clinical workflow that you can imagine run in the cloud using Amazon Rekognition and Amazon Comprehend Medical. So in this particular case, the use case is a clinical facility has this X-ray image that they want to de-identify so that it can be shared and put in a biobank and be shared with researchers for their use, right? So you can put all the data in an S3 bucket, and then using Amazon SageMaker, you use Rekognition to first identify all the text in that image, and then using Comprehend Medical, you can identify the PHI, the identifiable information in that text, and then basically de-identify it or remove it from the image, and then write it back in a de-identified form in S3 for researchers to use. Let's quickly talk about the various research initiatives. And I think this is where Dr. Norris also mentioned about the STRIDES program. So it's an NIH initiative that supports NIH-funded biomedical research in the cloud for researchers to get access to better tools, better training, and direct support engagement with the cloud service providers. It provides researchers, NIH-funded researchers, with cost discounts, enterprise level of support, subsidized training, consultation, reporting abilities on their cloud costs, and then access to an ever-growing NIH biomedical and genomic data repository. So a STRIDES program is a good example of where NIH and AWS are coming together to help build, sort of encourage the researchers to move to the cloud. The other program that I want to talk about, and I often get asked questions about it, is the AWS Cloud Credit for Research program, right? So this is a public sector program that AWS runs, which basically provides researchers, faculty, research staff, graduate students, postgraduate students, who have a specific need, a specific research project that they want to do on AWS, and they want to build and get access to the platform to do proof of concept or do benchmarking studies, or to help train the broader community. You can go to this particular link, and once again, I'll provide the links at the when the meeting with the recording of the meeting. You can go to this link, submit your particular use case, give a short description of your particular project, and get access to. I mean, it does go through an approval process, right? So there are subject matter experts at AWS who will help evaluate your particular project, and you can get access to AWS credits for your research. There are two other programs. We won't have time to get into those today, but I encourage you to also go look at AWS Educate, as well as Amazon Research Awards, right? Amazon Research Awards can provide the high-profile researchers in the field of data science and machine learning and artificial intelligence support to be able to do the next, to be on the cutting edge of science, to be able to do their projects in the cloud. And I think I'll stop there. I'm right on time, I think, and then take some questions.
Thank you so much for your presentation, Ankit. I learned a lot today. So we have a couple of questions I think we can go through. One of which, you mentioned numerous partners in kind of the public-private sector. So what are some key considerations when balancing accessibility, but also protection for privacy?
I mean, is this a question about, I guess I'm trying to understand, this question about access and privacy of data?
I
In which
believe
case,
so.
right. So, I mean, AWS works on a shared responsibility model, right? So AWS provides security off the cloud, whereas we are reliant on the customer, in this particular case, the research institutes, for the security in the cloud. We provide best practices for how you can secure your data, be compliant for HIPAA, for various other kinds of compliance requirements that the community needs for example, to be able to do clinical research. But it's up to the researcher to follow those best practices. We'll work with you, but this is the shared responsibility model. I hope that answers the question. I can't see the chat right now to see if
I believe so. That's all the context that I received. But I think you addressed at least how I interpreted the question. I think we have time for just one more. What advice would you have for students or trainees who are interested in pursuing this field?
Yeah, excellent question. So I was exactly where you are about 5 years back when I started hearing about the cloud, right? The advice I would have is start to look through, as we all do in science, start to read the papers that are starting to use cloud for their research. AWS has subject matter experts available. AWS has account managers available for each research institute, right? So if we want to get access to, for example, somebody who will answer your immediate questions, reach out to the account manager and I can help you identify who that person is. AWS also provides a wide array of training material for you to get started in the cloud, right? And then when it comes time for you to experiment, right? You're aware of the cloud. You have a particular way of doing things and you want to imagine how to do your day-to-day work in the cloud, reach out to the AWS research team or anyone of, or you reach out to me, for example, and I'll help you make that jump from being able to just use on-prem to versus reimagining how you do your work in the cloud.
Wonderful. I think that's fantastic advice for our young people. So thank you so much for joining us today and sharing your experiences. I would like to welcome Hatem Nawar, who is our next speaker in this flash series here. Natem Nawar is a Google Cloud customer engineer focusing on life sciences and genomics. In his role, he works with researchers and organizations, enabling them to use Google's capabilities in big data, analytics, and machine learning in critical areas of study and research. We're really excited to learn more about the Google Cloud application for computational genomics. And with that, welcome, Hatem.
Thanks, Kim. Okay, let me see if I can share my presentation. Okay, I see nods. Great, right. So my name is Hatem Nawar. I'm a customer engineer working with the public sector team. I'm based in London. So I work mostly with actually customers across EMEA and not the US, and I'm really happy to be here today. So let's start by this. This is actually a statement by Dr. Francis Collins back in 2005, that most drugs would be derived from our understanding of human genomes by 2020. And actually, fast forward to 2018, 19 of 59 drugs approved by the FDA were indeed informed by our understanding of genomics in the genome. Now, 2020 was, of course, a special year with all of the vaccine and the use of novel mRNA technology. This is even becoming more and more important. But what's more staggering as well is the amount of data that we have today. And Ankit, of course, and Dr. Norris talked about the amount of data that we have, not just genomics data, but imaging data, single cell imaging, proteomics data, and this different types of data that are available, which are exploding and making or introducing new challenges for how to process these types of data altogether. So a couple of quick notes from last year. Many organizations face different challenges. For example, looking at some of the projects I was involved in, some of the biobanks, for example, and some of the large genomics projects were actually struggling to have any data. Sample collection centers were not accessible because of travel restrictions, lockdowns, and medical staff being reassigned. And on the other hand, we had lots of variant surveillance programs, host sequencing, and vaccine development and approval programs. This shift can catch any organization off guard. And both the change in compute needs and the change in the type of compute required, as well as the amount of data, introduces challenges that can actually be addressed by some of the properties of cloud. So scaling from 0 to thousands. So if you are no longer have samples to process, you can easily scale down to 0 and keep your costs down. But if you need to access hundreds of thousands or even millions of cores, then you can actually do that. And then when you're done, you can scale back to 0 again. Another advantage is actually custom machine types. So you can shape the virtual machine or the compute resource that you're using for even every step of the pipeline. If you are running if you're running a variant calling, and then let's say you're running mark duplicates, it's a single threaded, you don't need a large machine. And then you move on to the next step of variant calling, for example, and you need a 16-core machine. So you can customize the exact amount of memory and the exact number of cores for each step of your pipeline. Finally, access to specialized hardware. And this includes, for example, large memory machines, machines that have GPUs, or even TPUs, which is Google Tensor Processing Unit, which is a special chip designed specifically to do machine learning. In addition to that, you have scalable storage, not just in terms of scaling to exabyte on Google Cloud Storage buckets, but also quickly provisioning high throughput storage on Google Cloud if you need a quick scratch space for your HPC applications, for example, and also, most importantly, access to both public data sets and controlled access data sets. So within Google, we have a public data set program. We have lots of genomics data in there, of course, including the 1,000 genomes, the Nomad, and others. But also you get access to controlled access data sets from the NIH through the STRIPES program or other organizations across the world. And this is actually key because when you look at something like the TCGA, it's accessed and used by thousands of institutes across the world, and just making thousands of copies is really not efficient. And we really need to actually consider our environmental impact of the research we're doing when we're actually doing this research. Then you also have the availability of actually building your infrastructure as code. So one of the recent projects we were working with one of the institutes, and they have transformed all of their infrastructure, which was a Slurm cluster, a DDN exascalar storage, and as well as some other infrastructure. This was all transformed into code. And whenever they start processing, in a few minutes, they actually deploy all of this infrastructure, which is secure because no one is actually building it manually. And once they're done, everything is simply removed. So because of all of this, researchers are able to do things that they were not able to do before. Having access to not just thousands of cores, but also special types of machines, like the latest GPUs or machines with huge amounts of memory, enables some of the researchers to actually do workflows and computations they were not able to do it before. So this statement from Dr. Peter Ashton from the University of York, we worked with them on one of the projects for water treatment and actually analyzing the metagenome through de novo gene assembly. And this particular workflow, they could run on any machine that they had available at that time in the university. It's simply every machine they would run, even when they run on a small cluster, would run out of me. So working with us, they actually used one of the UltraMem machines that allows you to use up to 4 terabytes. That was back then 12. Today we have machines up to 12 terabytes of memory to be able to run that particular workflow for the first time. And the benefit of that is you don't need to maintain such a specialized resource within your organization, but you can simply tap into these types of resources whenever you need to. So I'm going to go quickly through what I call a bioinformatician toolbox on Google Cloud. So lots of the developers of tools have been adopting cloud for the past few years. And what that means is there is a multitude of tools that you can easily access on Google Cloud. So as an example, one of the services on Google Cloud is Dataproc, which is our managed Hadoop and Spark clusters. And you can easily deploy Hail on top of that. And we're going to talk a little bit about that later. Another thing is workflow engines. And many of our pipelines now use workflow engines such as Nextflow, Cromwell, or SnakeMake. And at Google, we have actually built special APIs, which is life sciences APIs, that integrate with these workflow engines and allows them to run smoothly and make such a deployment actually quite easy to do for anyone with very little cloud background and technical knowledge. Finally, another vertical is actually the AI platform, which allows you to build machine learning models easily, gives you access to lots of the tools that we have developed at Google and BigQuery, along with Variant Transform. And again, we're going to talk specifically about this one. Finally, the raw compute. And sometimes this is really all what you need. Sometimes all what you need is simply access to compute and storage and networking to actually run your Slurm cluster or run Galaxy or run any of the other analysis tool that you want to run. In addition to that, some of the analysis platforms such as Terra that was co-developed by the Broad Institute and Verily also can run on top of Google Cloud, providing an easy to access workbench to run your notebooks, your pipelines, and also access to lots of data that's already on the cloud. And many more. So Hail is one of the tools that are used a lot. And Dataproc basically enables you to create Hadoop clusters in seconds. But it also enables you to easily scale up and down this cluster. So you can run a cluster with just a couple of nodes, and then on demand, you can actually scale it up to hundreds or even thousands of nodes. You can also add preemptible instances, which are kind of instances that come at a huge discount, 80% discount, but can be taken away from you at notice if we need this back, and actually drive down both the speed of the execution up and as well as the costs down. And once you're done, you simply go and delete all of that. There is no more waiting for queues on the local cluster or be limited by on-prem capacity. And this actually integrates directly with Hail. So if you want to run, for example, Hail on Google Cloud, all you need to do is type 2 lines of code, and then you're up and running. And we've seen actually some papers that was published by some of the organizations we're working with that was, they actually were running against a tight time deadline, and they were able to meet it because using such easy and existing tools. Another tool I'd like to quickly cover is the Variance Transform tool. And the Variance Transform tool is an open source tool that we developed that allows you to export variant data from VCF files into BigQuery. One of the challenges of developing machine learning models, especially for genomics and multi-omics data, is how can you get this data into a format that you can start to process using the common machine learning frameworks, such as TensorFlow and others. So with Variant Transform, you can actually take this, this variant data and actually quickly transform it, put it in BigQuery. You can then use SQL, and there is lots of easy integration with existing machine learning frameworks, such as TensorFlow and others. In addition, there are already lots of public data sets on BigQuery, like the 1000 genomes, the CCGA public access portion is there, and others. So this means you can actually integrate your data with the existing public data sets and process and actually analyze both quite easily. One of the examples actually for this, so Mayo Clinic has been building their own omics data platform, and they have used the variant transform to transform their variant data into BigQuery. Variant transform is also able to add annotation data, but what they did here is they've actually kept the annotation data separate from the variant data. This enables them to keep their annotations up to date, link that, and quickly analyze it. The power of BigQuery is extremely fast. And in addition to being extremely fast, it provides partitioning and clustering for the data. What this means is you can partition the variant data, for example, by location or by chromosome. So if you need to just query the variant data that you have from all of the sample, from thousands of samples, just for chromosome 13, you can do that, and it can be cheaper and faster, which is what they did. And of course, this all integrates with the strong security and access control management that's part of Google Cloud. Finally, looking at some of the workflow engines, and we've mentioned a few. So we've mentioned Promulps, SnakeMake, and Nextflow as some of the examples that already integrates with our Life Sciences API. So this is a sample architecture of how it would look like. So typically you would have your DNA sequencer, or if you're doing proteomics, your mass spec, or even if you're doing single cell imaging, could be your microscope. And then you push the data out to Google Cloud Storage through some of the existing tools that we have. And then you have a workflow manager. It's simply a single machine that have your workflow manager. Once you submit the pipeline, it starts spinning up the resources you need from the virtual machines, persistent disks, pulls the images from our container registry, and then once it's done, everything ramps down. So just to quickly show you how this pipeline or how this workflow works in cloud, let's say you have this as an example of a pipeline where you have one task, another task, then you scatter across different intervals of the genome, for example, and then you gather this data and start processing again. What will happen is you'll have one machine. Once the task is done, is going to be deleted. Another virtual machine is created for each task. And each one of these virtual machines could have a different spec. Some may have GPUs, some may have be a high CPU instance, and so on. And each time, once the task is done, the resource is deleted, so you don't have to pay for it anymore. This means that you can actually keep the cost down and make sure that you're using the right virtual machine for each part of your pipeline. An example of that, so the Finnish Institute of Molecular Medicine have been running the FinGen project, which is a very ambitious project that combines the genomics data from 500,000 citizens along with medical records and longitudinal studies and make them available to researchers. Now, the challenges of such a project is at some points in time in the life of the project, they would need to run a large analysis. For example, last year, they were running a GWAS for 180,000 individuals for 2,000 endpoints. And they needed to finish this analysis in a very tight timeline. So they actually burst to cloud. They used 100,000 cores and finished this in just a couple of days. Sorry, excuse me. And the interesting thing as well with cloud is the more you use it, the more you can actually save costs. So this is from the Broad Institute and how they managed to reduce the cost of the GATK best practices pipeline on GCP. So they started by moving the pipeline as is from on-prem to cloud. But then as they moved to cloud, they started realizing that there are lots of gains they can make. So they started looking at how they can do task splitting. How can they split the pipeline into multiple stages and use the right virtual machine, the right spec hardware for each part of the tasks. Then they started making use of the preemptive machine. Then they started using persistent disks. So instead of copying data from one machine to the other, they can simply detach a disk and then reattach it. And finally, we have worked with them to do further algorithm tuning. So working with them, for example, to get the HTS-GET library to read directly from Google Cloud Storage instead of having to localize some of the data or copying large sequence files or reference files to the virtual machine. We can directly read from Google Cloud Storage. So the end result is when they started, the cost of GATK best practice was around $47. Today, they have the $5 pipeline. So the $5 GATK pipeline, which does the same thing. And according to what I hear, is it actually costs just 3.65 cents sometimes. So as you can see, the amount of cost reduction is significant. And that's not all. It kind of shifts how you look at your costs, because you're no longer looking at the cost of purchasing large amounts of hardware that you may or may not use. For instance, we're working with one of the single-cell imaging projects, and for a certain duration, they're going to need lots and lots of GPUs. Now, this is what cloud can offer for you. You can suddenly gain access to hundreds or even thousands of GPUs to do the processing that you need without having to worry about the operational overhead and without having to worry about when you're done, what are you going to do with this hardware that you just purchased if you don't really need it. So not just that, but you're also able to put a price per sample. So when you're doing your budgeting, if you have an estimate for how many samples you're going to process, you can then easily say, all right, this is how much it's going to cost in cloud. There are lots of programs that we have to help researchers, our credit programs, our research credit programs, and our education credit program. I'm not going to go over them, but you can find more information about that online. And yeah, that's it. Thank you.
Thank you so much for that fabulous presentation. We do have a couple questions and we do have time before our next speaker. So our first question for you, Hatem, is how are platforms such as Google Cloud impacting remote patient support, especially during times such as these?
So we have actually, so not necessarily Google Cloud Platform, but actually the Google Workspace platform and our Google Meet. We have worked with healthcare organizations and actually published some open source repos to enable them to use our healthcare APIs that integrates with HL7 and FHIR formats, along with Meet, to actually create a telemedicine solution that can manage patient data securely and enable this kind of remote physician visit or medical healthcare. And there are actually many more applications. One of the interesting one was actually now with everyone is wearing a mask, people who actually relied on lip reading can no longer do that. So using our machine learning platforms, there are now ambulance services who has developed platforms that actually helps with using speech to text and actually allowing for further care. So there are some really interesting innovations that has came over the past year.
Wow, actually, that's a perfect lead into our next question here. So I'm sure you've seen kind of the evolution of this field as we're getting more versatility, more accessibility, lowering the cost and time for research in these types of questions. What new area or research question has surprised you or excited you the most with the benefits of Google Cloud?
I think one of the really great examples I can mention is what we've done, for example, with Moorfields Eye Hospital here in London. So cloud now offers lots of new services, especially machine learning, that I would say lower the barrier to access machine learning services. So what we have done is AutoML is a service that allows you to create machine learning models by simply uploading the data and labels that you have. And what we have done with Moorfield is, at the beginning, they had this collaboration with DeepMind, which is one of the Alphabet companies, to develop models that would detect certain eye disease. And then in the next phase, with very, very basic training, we've trained eye consultants to actually develop their own machine learning models without writing a single line of code. So one of the things that really excites me is with lots of the new services on cloud, we're actually lowering the barrier to access lots of the machine learning services to people who not necessarily has any coding experience. And they have actually published some of their findings, and this really excites me.
That's so amazing. We have time for just one last question. This comes from the audience. With your instances, is the state saved so the program can come back up easily with no cost or no loss of data?
This is so. I'm not sure I get the question. So when you shut down the instance, although the CPU and memory is gone, but you can still keep the disk. So anything on the disk is not lost. For our preemptive machines, it will depend on how the software is written, actually. So it's dependent on what you're running, what operating system, and what software you're running. So if you're doing machine learning with TensorFlow, for example, then there is checkpointing built in. There are some instances with BVM where that might happen, but yeah, it's basically dependent on what you're doing.
That makes sense. Well, thank you so much for this presentation and for sharing your experiences. I'm so glad that you could join us. So our next speaker for this series is Christoph Gorgula, who's a postdoctoral research fellow at Harvard University and is associated with the Harvard Medical School and the physics department. He develops and applies new computational and quantum physics-based methods for drug discovery. Today, Christoph will describe the development and utilization of a specific platform called Virtual Flow. Welcome, Christoph.
Thank you very much for the kind introduction, Kim. It's a great pleasure to be here today with you. So genes encode proteins, and they play a role in almost any kind of disease. So one prominent approach to cure diseases is to find small molecules which bind to the desired target proteins. And to find small molecules which bind such like specific target sites, which we see here in blue, like a pocket here, it's a great challenge to find really well-fitting molecules. So with traditional methods, we are facing several considerable challenges. Traditional methods like high-throughput screening can be quite time-consuming and expensive. The quality of the initial hit compounds can be quite poor in some cases. Also, if the target is more challenging, like if the target site is more flat, like in a protein-protein interaction, it's often not possible to find small molecules which bind tight enough to the target site. And if we find small molecules with phenotypic kind of screens, we have also the problem that often we don't know how the small molecule is actually binding to the target protein, or if it's binding to the target protein at all. These problems also affect the subsequent steps. So the first step in drug discovery is the hit identification, followed by hit and lead optimization, and then preclinical trials. And if the initial quality of the hits is, for example, poor, this can also affect negatively the subsequent steps. So if we could, in theory, screen dramatically more compounds in a short amount of time, in a more cost-efficient manner, this could make a big difference. And this is what we have tried with our new drug discovery platform called Virtual Flow. So here we see a typical high throughput library with 1 million compounds, which is already quite large because typically only a few 100,000 compounds are screened in a high throughput screen. Now here we see the space of the real library from enamine, which contains more than 1 billion compounds. So the small dot here in the middle, which you can see, it's kind of a drop in the ocean compared to billions of compounds. And Virtual Flow is able to screen this entire set of over 1 billion compounds, not only from the real library from enamine, but also other libraries like the Sync library. And this has many advantages. It is very fast. It can be done in a matter of a few days using supercomputers or large Linux clusters or cloud computing platforms. Also, it's quite cost efficient compared to high throughput screenings. And the quality of the initial hits is often remarkably good. So in our projects, we have been able to mostly identify big nanomolar compounds directly from the screen. And because we screen so many compounds and get more potent compounds, it becomes feasible to target a larger class of protein-protein interactions, which are one of the major avenues in the future also of drug discovery, and similarly for allosteric sites. Because the virtual screening uses molecular docking, we have a predicted docking process on how the molecule binds to the target protein. So we have also mechanistic insight or prediction on how it binds, which is also a big advantage. And this again affects also the subsequent steps if the first step here in inter-identification is improved. So Virtual Flow needs two major resources in order to be able to screen these large amounts of compounds. One is, of course, a large ligand library. And one of our primary libraries which we are using is the real library from enamine. It contains, so the version which we have prepared contains 1.4 billion compounds. And one big advantage is that these compounds are commercially available. So they can be purchased from enamine and can be delivered within approximately a month. And the second major resource is cloud platforms such as the Google Cloud, which we use mostly these days. And there we are using like 100,000 CPUs in parallel, where we parallelize the dockings, the docking instances. So now I would like to give you an overview of what Virtual Flow is actually doing. So here we see a target protein for which we want to find a small molecule binding to a certain site. So Virtual Flow needs here two main ingredients. One is the structure of the protein, the three-dimensional structure, which is usually obtained by X-ray, NMR, cryo-EM, and similar. Homology modeling is also an alternative in some cases. The second ingredient are the small molecules which we want to screen. So here, the space here represents actually the chemical space, and the white circles here on the top are ligands, small molecules, which are unprepared. In order to be able to screen them with Virtual Flow, we at first need to prepare them into a ready-to-dock format. So therefore, we have two modules of Virtual Flow. One module is dedicated to carrying out the virtual screening procedure itself, and the other module, VFLP, Virtual Flow for Ligand Preparation, is dedicated to preparing all these ligands. So yeah, VFLP, it does all the things like protonation states, tautomerization states, three-dimensional structure, and so on of the small ligands. Once prepared, here in the on the right, the violet stars represent the prepared molecules. once such a library is prepared, it can be used again and again for virtual screening, this virtual flow for virtual screening, VFVS. If we screen 1 billion compounds or more, we typically use a fast docking program, like QuickCleaner, for example. But if we want to increase the accuracy, we can do this, for example, by doing a rescoring procedure. Like, so we can take the top few percent of the compounds, like the top 1 million, and rescreen them again with higher accuracy. Here we can, for example, include protein flexibility, the side chain flexibility, backbone flexibility. We can use different docking programs in consensus docking mode or ensemble dockings if the protein is very flexible. If we want, we can even do a third stage where we use free energy stimulation methods, possibly also quantum mechanics if needed. For example, if it's a metalloenzyme, it can be important. At any stage, we can go to experimental validation, if we can order the best few hundred compounds and test them in the lab. If we have a compound experimentally verified, which is promising, but which we want to improve further, we can again take that molecule as a lead compound, go to the chemical space, search for the most similar, let's say 100,000 compounds called analogs, create a custom analog library, and then again screen it with virtual flow. And this cycle can be repeated if needed. Just to give a short history of virtual flow. So in ultra-large virtual screenings, one of the pioneers are actually Cat and Irwin from UCSF. They have prepared a sync library which for the first time provided hundreds of millions and now even 1 billion compounds for virtual screenings. In my PhD, I had studied it also, the topic a little bit theoretically, and we found, for example, that the true hit rate of the top scoring compounds increases with the scale of the virtual screening. In 2019, Cat and Irwin had a Nature paper where they found experimentally the same, that with the scale of the screen, the compounds get more potent and the true hit rate improves. And that also led to a news-infused article, "The bigger is better" in virtual screens. And they were also able to get directly a picomolar binder from their screen. They also had a second target protein, MC, and they also got directly nanomolar hits from the screen, from the virtual screen. Yeah, and there's another success story also from Nature, which also directly got a picomolar hit from the screen, this time the melatonin receptor. Our paper came out in the beginning of last year, where we published Virtual Flow as an open source platform. So it's free for everybody to use. And we, for the first time, targeted a protein-protein interaction. That's the keep one NRF2 interaction. And we also got low nanomolar hits directly from the screen, and also got compounds which are able to displace the protein binding partner. Most recently, last year, we applied virtual flow to SARS-CoV-2. So we selected 17 different proteins for which the structures became early available, and selected 40 different sites which are important for the virus, and screened 1 billion compounds each. And we got also nanomolar compounds for a number of these sites. And this is one. So here we can see that this is the NSP12, the polymerase complex from the coronavirus. And there are several protein-protein interactions here, like NSP12 interacts with NSP8 and NSP7. And we have targeted most of these interaction sites, both binding partners. Our paper about this was published in iScience a few months ago. And we also got the Reader's Choice Award together with Google, because Google was so generous and provided us with the required computation time for this project. So what do we actually need in the cloud, the Google Cloud, when we want to make virtual flow run? There are two main ingredients which we need. One is an elastic file system, which is a very high performance NSF file system. And the second thing which we need is Slurm, which was also just mentioned by the previous speaker. And Slurm we connect to Elastifile as the shared file system. And then this platform basically is the same, like the Slurm cluster is the same as is run on most supercomputers and Linux clusters from universities. And on that, one can run almost any program which was designed for such workload managers. And therefore also virtual flow. Because virtual flow was initially also designed to run on Slurm and similar workload managers in the academic setting, but because it's readily available in the cloud, not only Google, but also AWS and Azure and similar, virtual flow directly runs on these cloud computing platforms. We have tested the scaling behavior of virtual flow up to 160,000 CPUs on the Google Cloud. And one of the nice features which we can see here is that the scaling behavior of virtual flow is virtually linear. And the key for this is that the individual worker threads, which are working in parallel, they do not communicate at all. But we are using an advanced task list approach for this instead. And with that, I would like to thank you very much for your attention, and I'm open for any questions which you might have.
Thank you so much for your fantastic presentation. We have a couple questions already from the audience. So the first one, what is the advantage of using virtual flow compared to other platforms such as MOE or Schrodinger?
That's a good question. So with MOE, I don't have much experience. As far as I know, they are not yet at the billion compound scale, but I could be wrong. I didn't follow it very lately. And one of the major advantages of virtual flow is the scale, that you can really screen over 1 billion compounds, which makes a big difference in virtual screening. Regarding Schrodinger, yes, they also provide virtual screening methods. I'm aware, for example, that they also allow to screen over 1 billion compounds using their shape-based methods. But yeah, shape-based docking is a little bit different. It can have certain advantages when the shape is well defined. Yeah, but it's hard to say much more from me to these competitive platforms.
Lots of different use cases and applications for sure. So I think I may have missed this in your opening about virtual flow, but I was curious for the molecular dynamics simulations that you're doing for the actual docking. Are you using QMMM type of measurements for the actual ligand protein interaction?
Yeah, that's a good question. So we can use these simulations basically in two different steps. The first one is the structure preparation step, where we prepare the protein structure. And the second step would be when we do like free energy simulations to refine the list of hits in the end, like the third or the second or the third stage of the virtual screening. And these free energy simulation methods, they are not part of virtual flow, but you can use them in combination with virtual flow.
I see.
Yeah, because there is a lot of packages already available for these MT simulations, and you can just use the latest and what fits best to your specific target protein in use case.
That makes sense. Yes. So perhaps this also lends itself to the next question, in that are there particular ligand size constraints or protein size constraints? I saw one of your use cases had a GPCR on there. They're pretty massive in terms of kilodalton size. So do you have any comments on limitations?
Right. Yeah, this is a common question. And so basically, there is no restriction regarding the size of the protein. So there are two possible docking scenario cases which affect the size. One is a local docking, where on the protein, we know which site we want to carry out the docking procedure, like the target site. And in this case, we define a docking box or the docking sphere on that site. And it doesn't matter how huge the protein is otherwise, because the docking is refined to that box or the sphere. The second case is that if we don't know the target site, but we want to do a blind docking against the entire protein, then we can do something called blind docking. And we also have a docking program supported by Virtual Flow, which is specialized on blind dockings. That's QWinnerW. And this can screen the entire protein. So I have tested it with pretty large proteins. So GPCRs should not be a problem at all. But I'm not sure about the largest mega proteins. I haven't tested it with super large proteins.
Mm-hmm.
So, but yeah, the size should not be a problem.
Yeah, it's fascinating. I love the versatility and applications of these really exciting platforms. We have another question. What is the impact of using virtual flow on false positives in screening?
Yeah, false positives. I hope I get the question right. False positives are indeed a major problem in virtual screenings. So in typical virtual screenings, which involve a few million compounds, which I also did in the past, the success rate, like the true hit rate, is typically a few percent in my experience. But with the scale of the screening, like if you scale up the virtual screening with the number of compounds, the true hit rate actually improves. This follows theoretically by a statistical model, but also Scholket and Urban have shown this experimentally in their Nature paper. And the reason is simply that the more compounds you screen, the more better fitting compounds you are getting in the end. And if you screen like 1 billion compounds, the top scoring compounds, the best fitting compounds fit so well that even if there are some errors in the dockings, the fit was so good that it's still a true hit in reality, because it can compensate for some of these errors because the fit is so remarkably well. I hope this answers the question.
I see. Yes, I'm seeing in the chat that answered the question. So thank you so much for addressing that. Well, I think that's all for the questions that I see coming in live at the moment. I'd like to take this time to thank you for your presentation and thank the rest of our speakers for taking time out of their day and explaining these fabulous platforms. This is obviously a very exciting time for computational genomics with the versatility and accessibility that we heard about today. So now we'll take a short break and return for the next session, which is a funding panel discussion with speakers from NHGRI, DoD, and IQT. Thank you for tuning in for our session today.